Hi there, my name's Annabel Smith. I'm talking to you today from Brisbane in Australia, and I'm a lecturer in wildlife management here at the University of Queensland in Australia. And before this, I was a postdoc in Yvonne Buckley's lab at Trinity College Dublin, and I wanted to talk to you today about the work that I did there to give you a background on some of the population genetics work that we did for PlantPopNet. So just bear with me while I share my screen now. So what I wanted to do with this talk is basically give you an update of the published work that we have already done in regards to the population genetics for PlantPopNet. And the idea is that hopefully this will bring you up to speed with the data that we already have and the analysis that we've already done. So hopefully for those of you who are new joining the network or starting new add-on studies for PlantPopNet, we'll have a better idea of what's available and how that might be able to help you in your own work. So like I said, I'm a lecturer in the University of Queensland, and I live in a suburb called Indooroopilly in the western suburbs of Brisbane. And the Plantago here are currently in full flower, and this is one that I took a picture of just last week. So we have plenty of Plantago here, and there are a couple of PlantPopNet sites in this area as well. I've been here for about 2 years, and before that, like I said, I was a postdoc with Yvonne Buckley at Trinity College Dublin, and the work that I want to show you today was published last year, and it's based on the work that I did over a course of about 3 years with Yvonne and the PlantPopNet team. So this work we published in PNAS called Global Gene Flow Releases Invasive Plants from Environmental Constraints on Genetic Diversity. And it was based on the first 1 or 2 years of demographic data from the PlantPopNet network. So a lot of the demographic data we used in this study were actually those very early sites that were set up early in PlantPopNet that was already had data coming in from 2014, 2015. So some of the more recently set up PlantPopNet sites were not included in this study because we didn't have the data then. So this just shows you on the map those sites that we did use in this study. There were a total of 53 populations from 21 different countries. 35 of these were from the native European range, 18 were from the non-native range, and 44 of those sites were official PlantPopNet sites. So most of the populations we used in this study had matching demographic and genetic data. And then we had an additional 9 sites with genetic data only that we sampled specifically because there were either gaps in that sampling range or we really wanted to know something extra about those sites. Greece, for example, our preliminary analysis showed a really sort of strong structure in Europe, but we had no information from Greece. So Alan Finn and myself went down there to get some Plantago lanceolata samples and eat some Greek salad while we were at it. So there were a total of 554 individuals in this part of the study, and we used a total of 37,000 SNPs, so quite a big genome-wide genetic data set. The latitudinal range in absolute terms, so not thinking about north or south, was 27.5 to 61.4. So quite a wide latitudinal range across the global range of the Plantago distribution. And the paper had 48 contributing authors from the PlantPopNet network. So the first thing we did when we started to be interested in the genetic structure of the populations and what was influencing the genetic structure, and particularly how that related to demographic rates, because that's really the core of the, the strength of the PlantPopNet data is the demographic data. And we wanted to explore those relationships with between the demographics and the genetic structure. So what then affects genome-wide genetic diversity? And our first step was to put together a conceptual model to form the framework for the analysis that we wanted to do in this study. And we know from theory and data that demographic performance has a big influence on genome-wide genetic diversity. And in general, larger populations have greater genetic diversity than smaller populations. And this effect occurs through the effect on effective population size. More fecund populations as well tend to have higher genetic diversity. And there was a lot of evidence, empirical evidence to support those theories. So that's one of the primary pathways through which, one of the primary determinants, I guess, of genetic diversity. And then the environment comes into this in the sense that that affects demographic performance. So, we know a lot about, from spatial landscape genetics, we know a lot about how the environment influences population genetic structure and spatial genetic structure. But one of the primary pathways by which that happens is through that influence on demographic rates. And then, of course, there's dispersal. And now we know that dispersal has a huge impact on genetic structure. And this also affects genome-wide genetic diversity. The environment can affect genome-wide genetic diversity through this dispersal pathway. So that's one other pathway through which we can see environmental effects on genetic diversity. And this can happen via what we call natural pathways or human activity pathways, where humans sort of contribute to those ongoing dispersal patterns in plants. And there's also relationships between dispersal and demographic performance, of course. Demographic performance is going to affect dispersal and vice versa. And also the human influence. So when we transport plants to a new area and we get introduced plants or invasive species, they can also have that human activity dispersal can also affect demographic performance. So embedded in all these natural pathways, there's all these extra effects on top of that are influenced by human activity. We've got climate change, of course, affecting environmental conditions, which is going to have flow-on effects on genetic diversity through these main pathways. And then we have mass dispersal of propagules and cultivation, which affects dispersal. So that's the framework that we're dealing with here. And we put this together based on theory and based on data in order to then set up our hypotheses about what we expected to find when we analyze these relationships between genetic diversity and demographic performance in Plantago lanceolata. So we had 3 hypotheses that we tested in this study. And the first was that in the absence of dispersal, increases in survival and fecundity will drive increases in genetic diversity. So that's just that demographic performance genetic diversity pathway that we see here. And we thought that these effects will be diluted by dispersal between populations. So this should be, if dispersal is not restricted, this should be the primary determinant of genetic diversity. That the more dispersal you get, the less important this demographic pathway is going to be. The second hypothesis was that patterns of spatial genetic structure among native populations will reflect dispersal limitations across environmental gradients. And then we expected that in the non-native range, gene flow arising from multiple introductions will disrupt spatial genetic structure observed in the native range. So that's where we're seeing that human activity pathway come in. And then our third hypothesis was that environmental influences within population genetic diversity will be explained by demographic variation. So density, fecundity, and empirical population growth rates, which is the data that we had from the PlantPopNet network. So we're looking to see here, what we're saying is that we think the environment is going to influence genetic diversity, but we think it's going to happen through this demographic pathway, or we think we should be able to detect that through this demographic pathway. And then we thought that repeated introductions in the non-native range and long distance dispersal by humans would weaken that relationship, because they may be getting swamped with propagules from that increased human activity. So that's one of the major contributions of this study was that we had these matched demographic and genetic data sets, and it's at a global scale. And so a lot of what's been done on spatial population genetics has inferred those mechanisms that occur in the middle here, but we had the data to actually test these hypotheses more directly. So let's have a look at each of the hypotheses then and see if there was any evidence for them. The first one we set up to just test whether in the range of demographic parameters that we see in Plantago, would there be enough variation in this species to detect these kind of demographic only effects on genetic diversity? So what we did to test this hypothesis was set up a simulation experiment, and we parameterized the model with data from the PlantPopNet network. I'm not going to go too much into the parameters here, because if you're interested, you can read it in the paper. But basically, we parameterized a low juvenile survival scenario and a higher juvenile survival scenario. And we looked at the influence of female fecundity and that difference in survival rate on genetic diversity, which here we measured as heterozygosity. And what we found was that there was an effect of female fecundity at the lower end of that sort of realistic spectrum that you would find in Plantago, and that when female fecundity was very low, populations were most likely to go extinct. And then there was a sharp increase at around 20 seeds per plant. So, and after that, after about 20 seeds per plant, there wasn't much variation in the effect of female fecundity on genetic diversity. But we did see quite a strong effect of juvenile survival. So the high juvenile survival scenario had higher genetic diversity quite consistently than the low juvenile survival scenario. And these populations were not connected by dispersal in this simulation. So this is just looking at the effect of female fecundity and juvenile survival. And we can see these detectable differences, especially with juvenile survival, on genetic diversity. So what we're saying here is that in the absence of dispersal variation, we should be able to detect genetic variation related to the demographics of the population. And the second part of that hypothesis was that these effects would be diluted by dispersal between populations. And that's certainly what we found. Up to about 50,000 migrants per generation, we found differences in those two juvenile survival scenarios, but beyond that, there were no differences. And in this instance, migrants per generation was the migrants before recruitment. So it can be quite a large number because not all of those migrants are going to recruit. So that part of the hypothesis was indeed supported based on realistic variation in Plantago lanceolata. The second hypothesis was that patterns of spatial genetic structure among native populations will reflect dispersal limitations across environmental gradients. And what we did to begin with was just do a genetic structure analysis. So we're looking at, without putting any spatial information into the model, we did a structure analysis to look at where that genetic variation would lie based on the genotypes alone. And sure enough, we found quite strong spatial genetic structure in Europe with excuse me, with these different colors representing the different genetic clusters. And you can see on the plot here that Northern Europe tended to cluster together, UK and Ireland clustered together, Germany was more related to Northern Europe, Romania and Hungary tended to be quite unique, and Greece as well was more related to Romania and Hungary. So we're seeing this fairly strong spatial genetic structure in Europe that actually related to the spatial location of samples. And when we first did this analysis, it was all, you know, we could do a PCA without any spatial information, and it was almost like plotting the populations on a map. The genetic differences were really quite clear, and you see that within regions as well. So there's quite strong spatial structure. And the second part of that hypothesis was that in the non-native range, gene flow arising from multiple introductions would disrupt spatial genetic structure observed in the native range. And what we found was that most of the native populations were surprisingly similar. So across North America, one population in South Africa, a population sampled in Japan, Australia, and New Zealand were all quite similar, with New Zealand having some of this purple cluster here, which we think was probably related to some of the cultivars, some of those bred lines that were used in pasture. And there's more in the paper about that if you want to have a bit more of a look into that. So, but surprisingly at that global scale, those non-native populations are all admixed. So they contain bits of the genetic clusters from the European range, and they're relatively similar at that global scale. So multiple introductions, we also found that multiple introductions were required to produce the observed levels of genetic diversity in the non-native ranges. So we did another analysis that was looking at the minimum number of propagules that would be required to produce the kind of genetic diversity that we saw in these results. And we found that the minimum number of colonizing propagules required to produce the observed level of genetic diversity in non-native ranges ranged from 5 in New Zealand to 49 in North America. So just keeping with that same hypothesis for a minute, we also looked at FST, that's a measure of genetic differentiation between all pairs of populations in the data, to see whether they were related both to geographic distance and to environmental variables, which we did with a type of model called generalized dissimilarity modeling. We found there was quite a strong effect of geographic distance, but only when we looked at just the native range. So the figure that we're seeing here is data just from pairs of populations within the native range. There's this strong effect of geographic distance, such that populations that are far away are more genetically different to populations close together. So that's the isolation by distance effect. We found an additional effect that was not related to any of the other terms in the model of the seasonality and precipitation. So the genetic differentiation between all pairs of populations was stronger among populations in the native range than the non-native range. Genetic distance increased with geographic distance, and sites with similar levels of precipitation seasonality were more genetically similar after accounting for the other variables in that model. These results support our prediction that multiple introductions from diverse sources, diverse source populations and long distance dispersal can weaken the environment genetic structure relationship, because we didn't find this in the non-native range, or we didn't find it either in the non-native range or when we looked at all the data together, only when we looked at Europe only. And that brings us to our third hypothesis then, which was that environmental influences on within population genetic diversity will be explained by demographic variation. So remembering back to that first conceptual diagram, we're trying to look at that that demographic pathway through which the environment affects genetic diversity. And in this case, what we did find was that the environment affected demographics, but we didn't find a direct effect of the demographic rates on genetic diversity. So we had populations, there were additive effects of these environmental variables and range on population growth rate, with higher temperatures tending to have a lower population growth rate, and warmer sites tending to have a higher fecundity. So demographic variables are responding to environmental gradients, but they didn't induce a response when used as predictors of genetic diversity. So we didn't actually find that part of the hypothesis was supported. But the other part of that hypothesis was that repeated introductions into the non-native range and long distance dispersal by humans would weaken any relationships we saw in the data. So looking at the neutral genetic diversity, we found the similar effect in the native and non-native range of temperature seasonality on genetic diversity, with more seasonal sites being more genetically diverse. And in the native range, we saw a negative relationship between precipitation and genetic diversity, so wetter sites having lower genetic diversity, but no effect in the non-native range. So there's something going on here with neutral genetic diversity. We see that pattern in the native range, but not the non-native range. And similar patterns were detected, or even a bit more strong in adaptive genetic diversity. When we separated that part of the genetic markers that were putatively under selection, we found that in the native range, there was a strong positive relationship between temperature seasonality and genetic diversity, but not in the non-native range. A negative relationship with precipitation and not in the non-native range. So there's things going on in the native range that aren't happening in the non-native range. And so these results suggest that non-native populations are not constrained by the same environmental forces as their native counterparts. So then, thinking about all those hypotheses back in terms of the diagram we started with, our conceptual framework, we found that genetic diversity in Plantago lanceolata seems to be shaped predominantly by temperature and precipitation gradients related to gene flow and admixture rather than demographic performance. So we're kind of more finding this pathway through the dispersal, the effect of the environment on genetic diversity, we think is happening predominantly through that dispersal pathway. So high dispersal appears to have diluted any demographic effects on genetic diversity, and our simulation models showed that this would happen in this species. So genetic diversity in the non-native range is shaped by admixture from multiple source populations and ongoing introductions, leading to high neutral and adaptive genetic diversity in that non-native range. So it seems that from these data, invasive populations can establish in a broad range of environmental conditions without the need for associated demographic change. They're not constrained by those same forces as they would be in their native range. So that's just a background to the work that we've done and published already. And I guess I just wanted to provide that as a sort of background to get everyone up to speed who maybe hasn't seen this paper yet, and maybe to sort of think about how these data might help your own PlantPopNet project. Because I think what we've done so far is it's really just a starting point, I guess, of what could be done with PlantPopNet genetics. And when I was at Trinity, we had a lot of ideas coming out, a lot of new add-ons and different projects stemming on from this work. And I think there's even more going on now at the PlantPopNet headquarters there. So there's a lot more to be done with these data, and I'm sure that they're going to be useful in more PlantPopNet projects in future. So I'm really happy to help out where I can and to take any questions about this project in future. And thanks for listening. And I will be there at some of the PlantPopNet sessions during this virtual workshop. So I hope to see you soon.